again and welcome back to your biology practical classes. So this is your first biology practical class and today we will be going to talk about first about some basic rules of your biology lab and what you have to do for your biology practical copy and project. We will be checking the syllabus and after that we will be doing one practical. Okay. So at the beginning, let me tell you, as you are aware of that matter from the class 11, that at the time of entering into your biology practical lab, you need to wear apron. This is must. And the second thing is, you need to bring biology practical box. And these two you have to also bring for your examination also. Now come back to the next part. I will be telling you the syllabus of your term 1. As you know that your total practical syllabus and theory syllabus has been divided into two terms, term 1 and term 2. Under the term 1 they have provided some practicals and some other will be in the term 2. So first we will be going to talk about the syllabus of the term 1 and before that let me tell you about your practical copy and the project copy. As you can remember that for the class 11 also you have brought the practical copy from the school. The rule is same for this year also. So this is the biology practical copy of the previous year. So for this year it may look different, the color may change or the logo may change, but whatever you will be getting from the school, it is okay because this is the copy of previous year. I am showing you it as the sample copy. Clear? Now next and important thing is that you need to buy your practical copy from the school only. No other copies will be accepted which can be brought from the local market. So you have to buy and you have to submit your practical copy which is brought from the school and it looks like this. Okay, this is the previous year's copy and for biology most importantly you need to buy this threaded one, not the binded one, because in your school you will be getting two type of the copies, one is binded one other one is threaded one. So for the biology you have to buy this type of threaded one. What happened again? I didn't change anything. You can rejoin the call. Please rejoin the call. Now it is okay. Okay, so what I was telling to you that you have to buy this type of threaded copies and the number will be 2. Why 2? One will be used for the practical purpose and another one is for project purpose. So you need to buy this type of 2 threaded practical copies for the biology practical. One will be for the project and another will be for the practical. And most importantly, the pages also will be brought from the school. If anyone submits his or her copy within the channel file or within any other copies brought from the local shop, this will not be accepted. Why? Because you are going to appear for your board's examination. So this is important that everyone should use same type of the copy. That is why we are providing it, so that you can create a good impression and you can get good marks also in your board's examination. Clear? I hope all of you will be looking into that matter and they will buy these copies from the school very soon and will start their writing part. Okay, now come to the next part, that is about your project. As you can remember that in the class 11 I have told you different projects according to your name. This year also I will be following the same way, but not today. I will be providing you the project in the next class, okay? Because today I want to start this class with a practical. So what will be the project topics you will get, and what are the information or what are those different points you have to write down like the previous year. I will be telling each and every detail so that you don't face any problem at the time of writing your project. Clear? So this is all about the project. So now don't ask me again and again, ma'am, project. That means project I will be providing in the next class. Now come back to the points, the basic points of your biology laboratory again. First one, you need to wear white colored apron before entering the laboratory. Second, you need to bring the biology box which you have brought in the class 11. Third, you need to bring these 2 copies with you. Okay, and from where you can get, I have given all the information. Now come to the writing part. That means from where you will be going to write your practicals. So like the previous year, this year also you need to buy this book. What is the name of the book? Comprehensive Laboratory Manual in Biology Class 12. Comprehensive Laboratory Manual in Biology Class 12, and the publication is Lakshmi Publication. And the publication is Lakshmi Publication. Let me show you the front page also. Okay. So in the previous year you have brought Comprehensive Biology Laboratory Manual in Biology for class 11, but the same book you need to buy for the class 12. Okay, the class will be different. The publication and the name of the book is same. Comprehensive Laboratory Manual in Biology for class 12, Lakshmi Publication Limited. Yes? Ask me for what you have raised your hand. You can ask me. Clear everyone? So you need to write all the practicals which I will be showing one by one, and after showing all those practicals I will be telling that what will be those parts you need to write, because whatever is in your book everything will not be needed. Okay, I will be telling which parts are required, you will be writing according to that, and you will follow my words. Okay, what topic? Please try to join the class from the beginning, else you will not be getting what I was saying. Okay, now come to the next part. Next part is about your syllabus. That means what are the different marks distribution and what are the different topics are there in your revised syllabus. Now let me tell you. Just a minute. So from where you will be getting the syllabus, as you know that CBSE has released a revised syllabus, and at the beginning you will be getting the theory syllabus, and after that in particular syllabus you will be getting your practical syllabus also. So please check from that PDF which has been provided by the CBSE. Now let me tell you once. So for the class 12, there will be one major experiment which will be carrying 4 marks, one minor experiment which will be carrying 3 marks, and 3 spotting will be there which will be carrying 1 marks. That means 3 into 1, 3. 5, 4, 3, 3. And the total marks is 15. What about other 5 marks? Other 5 marks will be included in the practical copy, investigatory project and the viva, and all total 15 marks. As you know that your 30 marks is divided into, practical part is also divided into 2 terms, so 15 will be in the term 1. So this is the marks distribution of the each term. Okay, now let me tell you about the name of the practicals. So the first practical, which is a major practical, its name is Isolate DNA from available plant materials such as spinach, green pea seeds, papaya, etc. Term and second, or that means minor experiment, is prepare a temporary mount to observe a pollen germination. We will be doing this practical today. The first minor practical we will be doing today. And what about observation or study of the following or spotting? In the term 1, first you are having flowers adapted to pollination by different agencies like wind, insects and birds. 2, identification of stages of gamete development, that is testis, TS of testis, TS of ovary through permanent slides from grasshopper. Third number is meiosis in onion bud cell or grasshopper testis through permanent slides. Fourth number is TS of blastula through permanent slides, mammalian. And the fifth number is prepared pedigree charts of any one of the genetic traits such as rolling of tongue, blood groups, earlobes, widow's peak and color blindness. So, one major experiment, first experiment is the major one, which will be carrying 4 marks. Second experiment is the minor experiment, which will be carrying 3 marks, and the last, from all those spotting, you will be getting 3 spotting in your examination. Okay, so this is the total syllabus of your practical examination of the term 1 examination. Okay, so now what I have told you, that in the next class I will be providing you how you can write your project and the project topic, and today I am going to show you the first minor practical of your term 1, that is the pollen germination. Okay, so let me tell you about the theory portion of that practical. After that I will be showing that how you can perform this practical. Now before that, let me tell you one more thing, that at the time of your examination, you need to submit your project copy, practical copy, and an external examiner will come, he or she will take your viva also. So for getting this 5 out of 5, you need to prepare very well for giving all the answers at the time of your viva. So there is no as such syllabus for the viva, as you know that the topics which will be in your theory part and the topics which are included in the practicals and their theory part. That means practicals related theory part is also included in the viva. Questions may be asked from those parts. So whatever will be in the theory portion or in the practical portion, before the examination you need to prepare for your viva. So whenever I am going to show you any practical, I will be telling you in short their theory part, so that you can get a clear idea what and why we are going to do this practical. And on your examination day you need to write about some practical. So this will be your document, that what you have written. So at your copy please try to write down everything in detail, whatever I will be telling it to you. I will be telling you that how and what you have to write it down. You need to just follow my words for getting 30 out of 30 in your practical examination. If you follow every day's class and if you follow whatever I will be telling in the class, you will be getting everything to know. If you don't follow, then I can't help you on your examination day. You may do some silly mistakes, but for not getting a less mark than the 30, for getting 30 out of 30, please follow all the practical classes and whatever I will be telling you. Okay. Now come back to the practical. Today we are going to do this practical, and the name of the practical is prepare a temporary mount to observe pollen germination, and I am going to write it down on the board first. Please follow it. See the board properly? Please let me know if you can see the board properly. Now it is okay? Okay, very good. Give me 1 minute. So as you can see that project topic is already written on the board. So what is our topic? What is our today's topic? Today's topic is prepare a temporary mount to show pollen germination. Okay, we will be doing this practical. Before that, let me tell you about the pollen, what is pollen and how you will be doing this practical, and what are the steps we are going to follow. So as you know that pollens are very microscopic structure, right? And it is related with the male reproductive system or female reproductive system. Obviously you are aware of that it is related with the male reproductive system, right? So on which process, by which process the pollens can get formed, all this you have read in the theory portion, right? Still I am telling you once again so that you can get a clear idea about this practical. Now pollen grains are actually the microscopic and it is the first cell of the male gametophyte, and pollen grains can be formed by the process of the microsporogenesis. Okay, you have read this structure, their structure in detail. So the development of the male gametophyte is very precious. Okay, that means it begins in the inside or begins inside the microsporangium, or it is generally also called as pollen sac. Now you know that pollen grain sometimes at the beginning it is uninucleated, and after that, after some time, two nucleus will be there. That means it will be two-celled, and two nucleus will be there. One will be a small vegetative cell and another will be a large tube or generative cell. Okay, sorry. One generative cell and second one is the a large tube or vegetative cell. Okay, why these two type of the cells are present, what is their function, I will be telling you all this information after drawing the diagram on the board. Now what is happening, you can remember that after falling that pollen on the stigma, they will start to forming the pollen tube. Okay, they will start to forming the pollen tube. How? There will be an interaction between the pollen and pistil. Pistil will be releasing a sugary type of sticky type of liquid substance, through which their interaction, their conversation will be done, and these cells will be starting to form a tube-like structure which is called as pollen tube. Okay, that means that particular cells, tube cells or vegetative cells, will help in the arising of the pollen tube, and the another type of the cell which is called as generative cell, it is helping in the formation of the two male gametes. Okay. Now let me show you the structure of the pollen grain, and after that I will be showing that what is happening at the time of the germination. first diagram, can you see the board properly everyone? Please let me know. Everyone, please let me know if you can see the board properly. Okay, those who are saying the facing problem, it's from problem from your side, please rejoin the call. So, in the first picture you can see this is the diagram of the pollen grain. So, pollen grain is covered with some structure and its layers are told as outer layer is called as exine and inner layer is called as intine. That means we can say that pollen grain is made up of two layers, outer exine layer and the inner intine layer. And one more thing you are aware that exine is made up of most resistant substance which is called as sporopollenin. Okay, which is called as sporopollenin. Exine is made up of sporopollenin. It is the most resistant substance. It cannot get destroyed by any type of chemicals, pH, etc. That is why pollens can be viable for many days. They can survive in the adverse condition also. They will not get destroyed easily. But you can ask me, ma'am, if this type of hard resistant substance are there, then how these pollen tubes are forming? So for that you can see that there are some gaps. These gaps are called as germ pores where sporopollenin is absent. That means in the gas sporopollenin is absent and through these gas pollen tube formation will be taking place. And you can see two nucleus are there, two cells are there, and what is happening? A pollen is falling on the stigma and they are interacting with each other, are releasing a substance, sugary type of liquid substance, and this type of pollen tube formation is taking place, and this vegetative cell is forming this pollen tube and the generative cell is getting divided into two male gametes. And as you can remember that one male gamete is getting fused with the one female gamete or the egg, and another male gamete is getting fused with the central cells. So here 2 times fertilization is taking place. That is why we are calling it double fertilization. Okay, all this I have discussed in the theory class, right? Now you can remember. Please change your mode. Okay, this is the problem from your side. Please change the mode, make it in the grid view. So now come to that point. So here this is the another picture that what is happening. So this is the pollen tube and inside the pollen tube you can find two male gametes are there. Okay, this is the scenario what is happening. Now in your Viva questions may be asked that you are talking about the pollen, then what is the meaning of the study of the pollen? So it is called as palynology. Okay, it is called as palynology. The study of the pollen grains is called as palynology, and the pollens maybe have different type of the shapes. And now let me tell you what we are going to do in the laboratory. That means in the laboratory what we need to, how you can see this pollen tube formation. So for that, at the time of the writing, first you need to write aim. What is the aim? Aim will be the heading of the practical. That means to prepare a temporary mound to show pollen germination. This is the aim or objectives. After that you need to write down the requirements. So what will be there in the requirements? First one will be the freshly plucked leaves. Now this is the freshly plucked leaves. This is the freshly plucked leaves. Cover slip, slide, beaker, microscope, boric acid, sucrose, magnesium sulfate, potassium nitrate, etc. These are the main requirements for this practical. Now let's come to the procedure. So what you have seen that at the time of the in the nature what is happening, this pollen pistil interaction is going on and pollen tube is forming. But in the laboratory we will not be taking the pollen on the stigma. We will be doing it in the lab. So how you can do? You can make a nutrient solution which will be giving them the proper environment which they are getting in the nature, and by this we can be able to see the pollen tube formation. Now let me repeat that part. Then what will be the procedure? The first part or first point you need to do is the making of nutrient solution. Now what will be the different components that can be mixed and nutrient solution will be prepared? I am writing it on the board. Please note it down. These are the different components that can be used as the nutrient solution. So let me repeat. Please write it down in your copy. It is already written on the board. So first thing you need to do, you need to prepare a nutrient solution by dissolving 10 gram of sucrose. By dissolving 10 gram of sucrose. Listen carefully and you can write it down. You need to mix 10 gram of sucrose, 10 gram of boric acid, 30 milligram magnesium sulfate, 20 milligram potassium nitrate, which will be dissolved in 100 ml of water. Let me repeat. 10 gram of sucrose, 10 gram of boric acid, 30 mg magnesium sulfate and 20 mg potassium nitrate in 100 ml of water. So by mixing all these substances into 100 ml of water, we can prepare a nutrient solution. And on your examination day, I will be providing you this nutrient solution. No need of making, but examiner may ask you how you have made this nutrient solution. That time you need to say all these ingredients which you have mixed in the 100 ml of water. Okay. So first we need to prepare this nutrient solution, and after that you need to take few drops of nutrient solution on a clean side, and you need to dust the few pollen grains from the stamens of the mature flower, and after that you need to observe the slide under the microscope after 5 minutes, and then observe regularly after about half an hour interval. Okay, so this is the procedure of the practical. So first you are going to write objectives or aim, after that requirements, after that procedure, after that observation. So what you will observe in the nutrient medium, the pollen grain will be germinating. That means pollen tube formation will be there. That means tube cells emerges and comes out from the pollen tube through one of the germ pores and form a pollen tube, and the tube cell nucleus descends into the tip of the pollen tube, and the generative cell also passes into it, and it soon divides into two male gametes, and each male gamete is spherical type of, it is having spherical type of outline. Okay. So after the observation, on the left side on the white page you need to draw the diagram which is available in the book. Okay, you will get everything in the book. So just write down how I am telling you, and the last part of this practical is the precaution. So what precautions we should follow at the time of doing this practical? First one is flowers should be freshly plucked. Flowers should be freshly plucked, and second one is that you need to use the clean slides to observe the pollen grains. Okay, you these two are the precautions which you will be writing at the end of this practical. So this is all, that's all about the practical. Now I will be showing the procedure how you have to perform on your examination day. Okay, please follow properly everyone. So you can see this is the nutrient solution which already prepared by me. Okay, now let me show you. This is the dropper. This is the thin glass slide. It is clean. Now what we will be going to do, we will be taking few drops of nutrient solution on the slide like this. Few drops, 2 to 3 drops, and after that we need to hold the flower like this, and what we need to do is we will be doing like this so that some pollens can come into the on the slide. You can see, so which part is the pollen here? Which part? This yellowish colored part, right? So how you will be doing? You will be keeping the slide, and you will be holding that part, and you will be doing like this. After doing like this, some pollen grains will be falling on the nutrient medium which is kept on the slide. That's it. So now after that you have to keep the slide for few minutes. After 5 minutes you can check under the microscope, or you need to check after half an hour, 1 hour. You will be able to find out that slowly pollen tube formation is taking place. Why? Because we wanted to provide them the same type of solution which they are getting from the nature. We are making this nutrient solution to tell them, yes, this is the same type of the solution which you are getting from the nature, and in the laboratory we can see the pollen tube formation very easily. Okay, if I get any permanent slide of this pollen tube formation, I will be sending the picture. I will be taking the picture from the microscope and I will be sending it to you. Okay, so now cleared? Is it clear to everyone? Please let me know. It's a very easy one, right? Can you do it by yourself? Yes or no? Yes, very good, because very easy one. Just remember the steps and what are the important points I have told you. You need to remember all those for your Viva also. So that's all about today's class. Now you can ask me if you are having any question, any doubts, whatever you are having in your mind, you can ask me. I will try my best to solve all of your problems. Yes, both will be in the threaded copies. Okay. Anything else? Okay, so as you are not having any doubts, so I am finishing my today's class here, and don't miss the next class. We will be doing the major practical in the next class, that means isolation of the DNA, and parallelly I will be providing you your project topic and how you have to write it down. Okay, every detail I will be providing it in the next class. So thank you and bye bye for today, and see you in the next